This is Meglaine. She has a brain and neurons that make up her peripheral nervous system. One day, Meglaine's bird Lucy attacked her viciously. The sensory neurons in her finger picked up this external stimuli and began to transmit this information to her brain through a series of neuron connections. This is a diagram of a connection between two neurons. Dendrites extend from the cell body, which attaches to the axon via the axon hillock. The axon leads to synaptic terminals, which release neurotransmitters in order to communicate with a postsynaptic neuron. This is a graph of a neuron's membrane potential over time. In this video, we'll be addressing 5 different stages in this graph. At step 1, there is a large concentration of sodium ions outside of the cell and potassium ions inside the cell. The ion channels corresponding to each of these two ions are closed. This allows for a very negative resting membrane potential of a negative 70 millijoules. During step 2, the threshold membrane potential of negative 50 millijoules must be reached before an action potential is triggered. In this stage, the stimulus opens only some of the sodium ion channels, allowing for depolarization as the threshold is reached. During step 3, more sodium ion channels open, allowing for further depolarization. The inside of the membrane becomes positive with this large inflow of sodium ions, and the new membrane potential is positive 30 millijoules. An action potential has now been generated. During step 4, many of the sodium ion channels begin to deactivate as a process called repolarization occurs. Potassium ion channels then open, allowing for potassium ions to flow out of the cell. The membrane potential steadily decreases and is kept around negative 70 millijoules through the use of sodium-potassium pumps. These pumps are a form of active transport using ATP to pump 3 sodium ions out of the cell and 2 potassium ions into the cell. This allows the neuron to keep high concentration gradients for both ions within the cell.
Step 5. Some potassium ion channels remain open, allowing potassium ions to flow along the gradient to outside of the cell. The membrane potential decreases to negative 80 millivolts as the process of hyperpolarization occurs. Sodium ion inflow in one part of the membrane causes the depolarization of neighboring parts along the axon. The action potential travels in one direction along the axon as potassium outflow causes repolarization. This process is repeated along the entirety of the neuron, allowing a signal to flow in one direction and pass on to the postsynaptic neuron. Along the axon, there exist lipid membrane layers called myelin sheaths. Myelin sheaths are produced by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. The gaps between myelin sheaths are called nodes of Ranvier. Action potential are only generated in areas where there are nodes. This allows for a process called saltatory conduction, where action potential rapidly spread from the node to node. When an action potential reaches a synaptic terminal, the membrane depolarizes, causing calcium ion channels to open and allowing an inflow of calcium ions into the cell. The increase of calcium ions causes vesicles holding neurotransmitters to diffuse with the presynaptic membrane. These neurotransmitters bind to a ligand-gated ion channel located in the postsynaptic membrane.
Tetrodotoxin is a poison produced by several pufferfish species. Tetrodotoxin blocks the flow of sodium via sodium ion channels, meaning an action potential cannot be performed. Tetrodotoxin acts as a cork in a bottle, binding to the channel and blocking any sodium from diffusing through the channel.